through my lab many years ago. He's a Duke graduate. He earned his bachelor's here at Duke 25 years ago. Is that right? Yeah. So in any case, he's gone on to do great things, and I use Nii as an example, not only in terms of how far he's gone, but how whole he is. And he has a site about whole man. And so it just brings me to give the reputation I give to all my junior colleagues. You want to go far, but you want to be whole. You want to, get involved in your community, have a hobby, fall in love, do all those things that, life's a gift, life's a responsibility. Do right by life, and but do well by science as well. So do well and do good. So Dr. Nii Addy is the Albert E. Kent Associate Professor of Psychiatry and Associate Professor of Cellular and Molecular Physiology, the inaugural Director of Scientist Diversity and Inclusion at Yale University. And he is also Director of the Faculty Mentoring Program for Minority Organization and Action and Expansion, Co-Chair of the Career Development Subcommittee on Anti-Racism. And he earned his bachelor's here at Duke in biology and has gone on to do stellar research in terms of especially nicotine research, but it's expanded beyond that and also expanded in terms of his activity in the community, the broader community. And so this is sponsored by CCN, DIBS, and also the Center for Study of Addictive Risk and Resilience and Center for Addiction Change. And Dr. Addy's talk today is addressing substance use and mood disorders with neuroscience, advocacy, and social justice tools.
Thank you so much for that introduction. How's the volume, by the way? Can you all hear me well? Okay. Not too loud, not too soft? All right. I'm also going to time myself. This is a tactic I stole from a friend, but just to make sure that I keep on task here. But definitely appreciate that introduction. It's definitely been nice to be back here on campus. Definitely a homecoming, as you heard. So it's been great to reconnect with folks, even to see folks who were here when I started as an undergrad. And I do have to say that it's nice to be back in a place where I feel old, where I've had my transformative years. And the time here really shaped me in a lot of ways. It's also a little strange for me to admit that it's been about 30 years since I first stepped on campus. So even with my first meeting this morning, I tried not to say too much, but I had to kind of pause. My first research experience was actually in the Bryant Research Building on the 3rd floor with Anthony Mancha, who used to be here before going to Chapel Hill and I go to DC. I did cell molecular biology. I had no idea what I was doing. I was like, this is not going to work for me as a career. So I'm totally lost. Obviously, things changed, so it's good to be able to be back here and to be able to reconnect so many folks. So appreciate you all being here this afternoon. This did not exist when I was here before, so that's a little bit weird to walk through and see this random kind of glass elevator shaft in the middle of the quad. But apparently, there's things that happen down here. So it's good to be here with you all. I definitely appreciate that introduction. Just for welcoming me back in a lot of ways. So this is a blank slide on purpose. So over the last few years, I've actually changed how I do my introductions a bit. And so instead of just jumping straight into the science, I usually like to start with this slide. So you may recognize a few folks on here. Laura, I don't know if you can see yourself in that picture. Different hair color back then. But I start with this slide for a few reasons. So one of the reasons is because this really kind of helps me center everything in community. And so I think that's really important, something I've always thought about as a PI. Obviously, you can see that we enjoy eating. There's a lot of different snapshots of us at restaurants here. Also a snapshot of us bowling together and things like that. But I think it's important for us to remember how we actually work together. And it's something I've tried to be even more intentional about, especially after everything that we went through in 2020, kind of in the depths of the pandemic. And the ways that we try to think more about not only how do we work together as a team, but how do we actually show up for each other? How do we support each other? And how do we navigate through? I think this is especially important, again, 5 years later at this point in time, for those of us who are in biomedical research with all the uncertainty that's happening right now, even as we're thinking about things like funding and support and students and trainees And irrespective of where anybody might fall across the political spectrum, this is a time when all of us know that there is uncertainty, when all of us are not sure what's coming next and we're trying to navigate through together. So again, community to me feels very important. And I think about this not only in terms of my lab community, but also how do we interact within our molecular psychiatry division or interact with folks in our psychiatry department or in the School of Medicine. Not just those pieces, but also at the university in general. Obviously, each of our universities are in local communities as well. So we also have to think about how do we actually engage with people in our local community or in the state community, the country, the world. And I know when I say that, it sounds a little bit like hyperbole or maybe too philosophical, but I think it really is important. And for me as a PI and as a mentor, it's really encouraging to see the ways that folks in my lab have tried to engage in that as well. And so the other thing that I've done in this picture is actually show some images of things we've been able to do in the broader community. So we've had town hall conversations in different parts of the country, in different states. We had one in New York City in 2019. Actually, a Duke faculty member, Warren Kinghorn, came up and joined for that. He's a faculty member here in psychiatry, also has an appointment at the Divinity School. And so to be able to engage with communities about aspects of neuroscience or mental health, work with advocacy groups, and to have conversations that people have really gotten a lot out of have been really important to me as well. So again, I say all that just to kind of center the research and put it in that perspective. The other slide that's always important for me to show beforehand is also this slide here. And again, you may recognize some pictures of folks here. Those who've been at Duke for a while may even recognize some for administrators. So the reason I show this is because, again, this represents people who have been mentors, sponsors, friends, collaborators, and colleagues over the years. And it's not an exhaustive list, but to me, this picture montage, as it were, is always very sobering and a great reminder. Some of the folks on this slide have actually passed, so that also gives me a sense of pause and remembrance as well. But of course, it includes folks like Dr. Levin, my undergraduate advisor, my grad student advisor, Marina Machado, my postdoc advisor, Mark Whiteman. But there are also folks here who are in the public space. There are community leaders here. There are pastors here. There are former university administrators. There are friends who started nonprofits. There are lawyers on here. And so again, it represents kind of a wide range of folks who've invested in me over the years. And so one of the reasons I think it's important to start here, again, sometimes as investigators, we have this tendency to talk about the work that we've done without always acknowledging all the people who have supported us in that work along the way. And as you'll hear, as the students have already asked me about today, the many things that I'm involved in, this is definitely a team sport. There's nothing that I do in isolation, but it's not only a team sport right now, it's been a team sport for years. All the people who've really invested in me over the years and continue to do that. So these are my centering slides. Hopefully, as I'm going through, you all can kind of think about some of those folks in your own lives as well. But with that, let me transition and now get to the actual talk itself. I will admit that the title that I've put here is quite ambitious. So addressing substance use and mood disorders with neuroscience, advocacy, social justice tools. That's a lot to try and put into one talk and a lot to try and put into one career. So I'm open to feedback. You all can critique as we go through as well, whether you think we're actually achieving this. But again, I think it really speaks to this importance of working amongst teams and partnering not only in our university settings, but actually partnering with others as well to move these things forward and to be able to make an impact. So the other thing I kind of want to do as I give this talk today is give you a little bit of a sense about how I talk about some of these things in general public settings. So obviously you all are an audience of experts in your respective fields, and these are things that you know about in terms of background. But I think it's also important for us to think about how do we actually communicate these types of things to the general public. So typically when I'm speaking to the general public, one of the main things that I talk about first is the fact that as a neuroscientist, I'm basically interested in the brain biology of complex behaviors. So why do we do what we do as human beings, and what is underneath that? So I always start there. I always also make the point for why do we actually do research, in my instance, in rats and mice. And so if you've ever come across any late night TV shows where there's been a neuroscientist that's on, the host will invariably make a joke about neuroscientists telling him or her, well, are you saying that my brain is just like that of a rat brain? Obviously, that's not what we're doing in science, but the thing that we always need to drive home is that there are enough similarities between a mouse brain, a rat brain, between primates and humans, that we can actually learn things from these animal models that can actually inform our understanding about the brain in general and also about human behavior. Now, obviously, these are things that you all know, but I think it's important to remind the general public of those things as well. So in terms of our research, once I've kind of set that foundation, I then let people know that we're not just interested globally in complex behaviors, even though we are, but from a research standpoint, we've just kind of zoomed in and focused on motivated behaviors. So from a very reductionistic standpoint, we are thinking about that in terms of positively and negatively motivated behaviors. So positively motivated behaviors, things that we might seek out that are good and beneficial. Negatively motivated behaviors, things that we might avoid. The next layer on top of that is thinking about those types of behaviors, particularly within the context of substance use disorders and mood disorders. So the other thing that I do, which I won't do here today, but when I'm speaking to a general audience, I'll actually have them share with me the types of behaviors that come to their mind when they think about substance use and misuse, and the types of behaviors that come to their minds when they think about mood disorders, so things like anxiety or depression. And invariably, people are actually pretty good at this. They'll very quickly think about certain behaviors that come to mind. What they'll also notice is that there's a lot of overlap between the two. So the types of behaviors they talk about for substance use disorders tend to overlap with those they think about for mood disorders, often around aspects regarding motivation. So that's always an instructive thing for me to have people say out loud. It also is a nice segue because what we've been thinking about in the lab is not only these as separate entities, but also paying attention to the fact that there's a lot of evidence showing that those with substance use disorders have a higher prevalence of mood disorders and vice versa. And so that's something that we're also paying a lot of attention to. So with that background, I'll tell you a little bit about some of the goals in our research. And one of that has been to try and understand the neurobiological basis of these behaviors in the first place, to understand what's meaning these behaviors. And we're thinking that that will actually potentially give us some insight into the actual etiology or cause of the disorders and illnesses themselves. But we don't just want to stop there. We're also focused on trying to actually identify new and effective therapeutic targets and interventions. So with that framework, I'm going to lean into some of the work we've done over the last few years. I'm going to start by talking about really the foundational work in the lab where we were looking at the positively motivated behaviors, specifically in the context of substance use disorders, but thinking about aspects of reward taking and reward seeking behavior. Then I'm going to pivot to some of our newer research looking at negatively motivated behaviors in the context of mood disorders. So looking at things like responses to stress, anxiety, anhedonia, avoidance, and pain motivation. And then I'll wrap up the scientific part of this talk, looping those two together and thinking about this active aspect of comorbidity. So before I get there, again, this is thinking in the context of what I would typically do for a general audience, but I think it's also important here. I want to talk a little bit about the models that we use. Now, a lot of these models are familiar to many of you because I know that work is already going on here at Duke, but I thought it'd be a good refresher nonetheless. So for us, we're particularly looking at drug taking and drug seeking behavior in rodents. And so we use the intravenous self-administration model, which has been around for decades, but we perform these surgeries in the house. We insert a jugular or a catheter into the jugular vein, place the animals in an operant chamber, and give them access to two levers. So every time they press the active lever, which in this case is on the right, they're going to receive an intravenous infusion of cocaine. And that's going to be coupled with this light, cue light above the lever that's going to be illuminated, and it's going to be an auditory tone that is also played when they press that lever. In this instance, every time they press the other lever, which is inactive, there's no programmed consequence. And what you see over time is the animals, as you would expect, are very good about discriminating between which lever produces cocaine and which one does not. And so as you can see here, over a 10-day training period, we're letting the animals press for 2 hours a day, and they do get pretty steady levels of cocaine self-administration over that training period. Now, one of the conversations I had with some of the trainees today, and one of the things that we're thinking about is whether we should still use this short access model or whether we'll start using some of the more extended access models and intermittent access models as well. That's something we can come back to in the Q&A session if people have questions. But for the purposes of today's talk, I'm going to be describing our work looking at cue-induced cocaine-seeking behavior. So after this training period, we'll take the animals through different periods of abstinence where they don't have any access to the cues and no access to the cocaine. This is basically similar as what you might see with detoxification, for instance, where you don't have any type of exposure therapy or extinction. So after a certain period of time, we'll place the animals back in the chamber. We'll again let them press the levers, except this time, every time they press the active lever, they're only going to get delivery of the cue that was associated with cocaine, but no cocaine itself. And so what you can see looking at different periods of abstinence is that the animals still press much more for the lever that produces a cue and not so much for the lever that does not. And so we use this as a readout of our cue-induced cocaine-seeking behavior. I should also point out this only occurs if you pair the cues and the cocaine together during training. If you have the cues presented randomly during training, you don't see this behavior emerge. Now, some of you in the audience are also interested in the incubation of craving, where you actually see that active lever pressing increase over time. Again, that's often seen with a more extended access training period and with higher amounts of cocaine. So that's not something that we've looked at here, but we have developed that in the lab as well. But for the purpose of today's talk, I'm going to be focusing on this cue-induced seeking, where we see pretty steady levels over time. So one of the things that we were doing early on was just to look in literature and see, well, how have people actually looked at this aspect of cue-induced craving and relapse? And really, this is going back to psychology and thinking about this aspect of stimulus reward learning, which I know is familiar to many of you already. But just to briefly highlight it, I've shown a few different pictures here on the left. Obviously, for that animal, they've learned to associate certain cues in that environment with the availability of food. I have no idea what that animal has in its mouth, but apparently it's tasty, and apparently it was motivating for the animal to actually perceive that and pursue it. For me, that image next to it is something that's very salient. You could argue that's just a random conglomeration of plastic and metal, but for many of us, that's something much more than that. I've had my coffee today, so I'm not having a craving right now. I always say when I show this, if people get up and leave the room, I always know why. I also am not asking you to self-disclose what cravings you might be having at any point in this talk with any of the images that I'm showing. But we know that coffee and caffeine can be a salient reward for many of us, and so that's a cue that will induce craving. If there were someone who uses substances or has a history of misusing substances, that can include visuals of those substances or people that we associate taking those substances with. So obviously this goes back to Pavlovian conditioning. We have the unconditioned stimulus, the reward, this pair with the conditioned stimulus. So often that the conditioned stimulus elicits craving for that reward. As you all may know, this is something that's not just specific to substances, but also to natural rewards. And there's also this idea that's in the literature for almost 20 years now, that the conditioned stimuli kind of in and of themselves become desired and wanted over time. So one of the classical examples I often talk about is if you talk to an individual who has a history of smoking, who has had learned to enjoy the smoke or the throat hit that you get in the back of your throat every time you smoke a cigarette. Oftentimes when I'm talking to people who are trying to decrease their cigarette use or quit, they'll say they miss that throat hit that they got every time they smoked a cigarette. So much so that they desire that. Now that throat hit itself is not anything that's reinforcing in terms of nicotine reinforcement, but it's become associated with smoking behavior so much so that people desire it. And it's one of the things that people have also talked about in terms of e-cigarettes, sometimes not feeling that same throat hit that they used to get with a cigarette and still missing that sensation. So with all that background, we were trying to think about, well, what have people actually done to look at what's happening in the brain that allows this craving to emerge in the first place? And so this is data that was collected almost 26 years ago now by Anna Ray Childress and colleagues, basically looking at brain imaging in individuals who were dependent on cocaine versus those who weren't, and showing them videos with cocaine-related cues or natural-related videos. And what they saw, not only with cocaine, but with other substances, is there was increased activity in dopaminergic regions of the brain when they saw cocaine-related cues and videos. So a lot of evidence suggesting that these processes, these dopaminergic regions were important. So early on in the lab, we decided to focus on specific regions within the mesolimbic dopamine system. So here I'm actually showing a sagittal section from a human brain, but focusing on the ventral tegmental area where the dopamine cells are located, and the nucleus accumbens, one of the regions where these cells project their neurons to. And there were a few reasons that we decided to look at this. Again, going back to the human imaging data, but also being informed by a lot of the work that had been done in primates by Wolfram Schultz, specifically looking at the firing patterns of these neurons in response to both rewards and cues. And what they found is that you have rapid or burst-like firing, high-frequency firing of these dopamine neurons when you present unexpected rewards, but also when you present cues that predict these rewards. And you actually see the same phenomenon in the nucleus accumbens when you look at the release of dopamine. So a high increase in dopamine in response to unexpected rewards in terms of dopamine concentration, or to cues that predict rewards. And so a lot of evidence showing correlations. There were also work that was done in our lab and others, showing that this dopaminergic activity is actually necessary and sufficient for some of these reward-seeking behaviors. So if you block this firing activity or release, you don't see this reward-seeking behavior. If you enhance it, you do see this reward-seeking behavior. So with that context, we decided to delve into mechanisms within the ventral tegmental area. There's a lot of information on this slide, which I'll go through in time, but basically to say, what are the processes that can actually modulate this dopaminergic activity? And can we actually modulate the behavior by modulating that dopaminergic activity? And so one of the first things we decided to look at was the nicotinic receptor mechanisms, again, going all the way back to my days in undergrad and for the foundation that Ed gave me in a lot of ways. But we also started to think about not only nicotinics, but muscarinic receptors. And then eventually we also started to think about L-type calcium channels as well. And so I wanted to share a story here because there's a very strong scientific foundation for why we decided to look at the L-type calcium channels. But that wasn't how it actually started. And so this, I think, is an important piece in terms of just thinking about and realizing the importance of networking. So when I first started lab, I basically went to a Gordon Research Conference on catecholamines, and I was introduced to an L-type calcium channel expert, Anjali Rajadaksha. The only reason I was introduced to her is because her graduate student at the time used to be a technician, a post-bac technician at Yale while I was a grad student at Yale. And she said, oh, you should meet my PI. So I met her PI. Anjali actually invited me to give a talk at Weill Cornell, where she was at the time. I went down there to give a talk, and we realized that we had overlapping interests. And so we actually started to think about collaborating. We did some pilot experiments. We had some joint lab meetings. That collaboration actually eventually led to an R21 grant, which we were able to successfully get on first submissions, to my surprise. That eventually led to an R01. So we've continued to collaborate over the years. And as I'll talk about later in the talk, that's actually now leading to clinical studies that we're actually getting ready to get off the ground. So I say all that to say that that wasn't my intention in going to that conference or in having that interaction, but just being present sometimes can be really important. And I say that for folks like me who might identify as introverts or folks that might identify as extroverts. Sometimes students will ask me, well, how do you actually navigate those spaces if you're not someone who naturally goes out and talks to people? Sometimes just being there is enough. I'll share another quick story about times when I didn't do that so well. So some of you may know we also do a lot of work around tobacco. At one point, one of the program officers for those grants as we were getting off the ground from NIDA, we were all at a conference, I think either the Society for Research on Nicotine and Tobacco or maybe a conference in DC. And I was talking to my program officer, and then someone very high up in the field came and started talking to her because of the project they had. So I basically just politely excused myself from the conversation and backpedaled my way out of that conversation. She actually got very upset with me later on. She said, why did you do that? I was about to introduce you to so-and-so. So again, being an introvert, I didn't have to do much. All I had to do was actually just stay present. And so I share that as a story to say, sometimes you don't have to actually go out of your way to have these interactions, but just being present in that situation can lead to opportunities. So with all that backstory, let me tell you a little bit about what we have started to do scientifically in terms of these L-type calcium channels. Well, it turns out that these channels are also very good at regulating dopaminergic activity in the ventral tegmental area in particular. So there have been physiology studies that have looked at this and found that if you activate these L-type calcium channels, you can actually induce that rapid burst firing in these VCA dopamine neurons that is known to be important for reward-seeking behavior. And there are other studies that have also looked at this in other ways as well. It also turns out that there are genome-wide associating studies that have associated gain-of-function mutations in these channels with different neuropsychiatric illnesses, including cocaine use disorder and bipolar disorder. The third piece is that L-type calcium channels are actually, or the L-type calcium channel inhibitors are actually FDA approved for hypertension. And so as we were starting to think about their involvement in these neuropsychiatric illnesses, we also realized that there's an opportunity to potentially repurpose some of these medications for some of these outcomes and behaviors that we're looking at as well. So with all of that, one of the first things that we wanted to ask was whether these L-type calcium channels, particularly in the ventral tegmental area, will regulate this seeking behavior, this cocaine-seeking behavior that I just alluded to. So in these experiments, what we did is to train our animals, again, to self-administer cocaine in the same way I described to you earlier. We then took them through 10 to 14 days of forced abstinence and then placed them back in the chamber and allowed them to perform this cue-induced cocaine-seeking behavior. Except that before that behavior, we infused the L-type calcium channel inhibitor directly into the VCA to see how that would impact the behavior. And so what we found was actually a very robust decrease in cue-induced cocaine seeking. So as you can see here, looking at the active lever, with the higher doses of isradipine, the L-type calcium channel antagonist, we were able to decrease their cocaine-seeking behavior. We also confirmed this with another L-type calcium channel inhibitor, nifedipine. So it seemed to be not specific to inhibitor, but specific to the L-type calcium channel inhibition. We were also curious if this was just a general decrease in reward-seeking behavior or specific to substances. So we trained another cohort of animals to go through sucrose self-administration and then sucrose-seeking behavior. When we blocked L-type calcium channels in these subjects, we had no change in the behavior. So it seemed to be very specific to cocaine-seeking behavior, and this is actually similar to what we had seen previously with nicotinic receptor antagonists. Now, I mentioned before as well that dopaminergic activity is causal and necessary and sufficient for this behavior. So we wanted to see whether this dopaminergic activity was also being regulated by these same manipulations. So in these experiments, what we did was to place a stimulating electrode in the ventral tegmental area so we could stimulate burst firing these dopamine neurons, and then put a recording electrode in the nucleus accumbens where we could measure dopamine release in real time using voltammetry. We also coupled that stimulating electrode with a cannula so where we could infuse pharmacological agents directly into the VTA. So again, we knew that dopaminergic activity was necessary and sufficient. So what we predicted is that if we had a decrease in the behavior, we'd also have a decrease in dopamine signaling. When we did the initial experiments, we actually saw the exact opposite. So these are animals that are in cocaine abstinence. We block L-type calcium channels in the VTA, we stimulate, and we actually see an increase in dopamine signaling compared to our vehicle animals. So my postdoc at the time was actually very frustrated when he collected this data because it was opposite of our hypothesis. He kept collecting the data, kept getting the same result, and kept being more frustrated. I felt like he was doing something wrong. So there's a story and a lesson there in and of itself. But one thing we realized is we said, okay, well, we also have these animals that are sucrose abstinent and don't have this behavioral change. Let's see what happens if we block the L-type calcium channels in those animals and look at dopamine. No change whatsoever. So at this point, I should admit we were still stuck on our hypothesis. We said somehow dopamine has to be decreasing. Obviously, that's not a good framework to use, but that's where we were. So we also knew, we thought, okay, well, we're looking at this rapid burst firing. What if we look at tonic firing of these neurons, which also happens in vivo, 5 to 7 hertz? Maybe if we tonically stimulate these neurons, there's a change in dopamine signaling that would fit our hypothesis. Maybe there's a change in signal to noise where we have this rapid increase with phasic responses, a different type of increase with tonic responses, and we still have an overall decrease in dopamine signaling. Obviously, there's a lot of problematic aspects with how we were thinking about this, but I'm giving you all this background in terms of full disclosure. So we did the experiments with the tonic administration and saw the exact same results. So then we thought, okay, well, if we're thinking about this in terms of therapy, in humans, this would have to be given systemically, so let's do this systemically and see what results we get. Same behavioral results, same electrochemical results. So eventually, we finally stopped being stuck on our hypothesis and we said, okay, let's actually figure out if this increase in dopamine is actually leading to this decreased behavior. So we had to be a little bit strategic in terms of setting up those experiments because as I told you before, if you enhance the dopamine system, you promote the behavior. If you block the dopamine system, you decrease the behavior. But what we were trying to do was to say, okay, well, it seems like if we block the L-type calcium channels, that's increasing dopamine release in the accumbens, which is acting at the receptors to decrease the behavior. So let's try and block the receptors and see if we lose that effect. But blocking those receptors themselves, as you can see here with flupentixol, already decreases the seeking behavior. So we actually picked a sub-maximal concentration of the dopamine receptor blocker and used that in the accumbens to see if that would impact the behavior. So just to walk you through, we again took the animals through self-administration, abstinence. We gave them an IP administration of the L-type calcium channel inhibitor 15 minutes prior to the test to give it time to access the brain. And then we used the dopamine receptor antagonist at this sub-maximal dose directly into the accumbens. So what we found is that in the animals, as we would expect, that went through cocaine abstinence, you do have this cue-induced cocaine seeking, much higher active lever pressing than inactive. If you block L-type calcium channels, you can inhibit that. If we give the sub-maximal concentration of flupentixol by itself to block the dopamine receptors, you see a modest decrease. But now if we add the L-type calcium channel inhibitor on top of that, there's no change compared to the flupentixol itself. So it turns out that that increase in dopamine was actually facilitating the decrease in behavior. But we weren't able to appreciate that until we finally got off of our hypothesis and actually tested what the data was showing us. So there's a lesson in there in and of itself. But something which we've seen, again, both with systemic and with intra-VTA administration. And so I'm going to come back to that in a second, but basically I'm going to pivot a little bit and talk about some of the work that we've done to also look at these same things in the context of mood behaviors. And again, as a behaviorist, I don't know if I can call myself that, but I'm going to for today anyway. I feel like with the foundation I had in Ed's lab, that's a very appropriate comment to make, or maybe behavioral pharmacologist is better term. But Ed can correct me if he likes as well. But we've also tried to think about, well, what's the best way or the best ways to think about some of these mood disorder related phenotypes. So several conversations I've had with program officers at NIMH over the years about how we can't call it depression in the rats and mice, which is valid. But we're also still trying to see, can we actually pull out some pieces of behavior in the rodents that will give us some insight into what's happening in humans. So we use a range of different behavioral battery of tests here. I'll just go through a few of them. One of the ones is the elevated plus maze. Again, some of you may be familiar with this, but as the name implies, it's a plus maze that's elevated off the ground about 40 centimeters. And what rats will do if you place them in the center, they'll tend to spend more time in the closed arms, dark arms, and only a little bit of time venturing out into the open arms. If you give them an agent that is anxiogenic or produces anxiety, they'll spend more time in the closed arms. If you give them something that decreases anxiety or is anxiolytic, they'll actually spend more time venturing out into the open arms. We also use a sucrose preference test. So the animals basically have the choice of choosing 1% sucrose or water. And typically, if you give animals access to this, they'll prefer sucrose anywhere from 60 to 85% when you compare between the two. If they've gone through stressful situations, they'll shift and not show as much preference for sucrose. And people use this metric of anhedonic related behavior. We've also integrated the marble bearing task. Marble bearing task here. So basically, there are 24 marbles in this cage with bedding, and rodents naturally will go and try and bury all those marbles. Again, if you give the animal something that's anxiogenic, they will bury more marbles. If you give them something anxiolytic, they won't bury as much. We've also set up the social interaction test in the lab as well, as I'm showing you on top right here. And basically, we place the animals in this center chamber and allow them to interact with either a novel rat or a novel object. And the animals typically show pro-social behaviors. Will spend more time with the novel rat than the novel object. We've also started to think about things like effort and motivation. So this is a paradigm that was pioneered by John Salomon called the effort-related choice paradigm. Basically, place the animals in the operating chamber and give them access to either freely available chow, or they can press a lever 5 times to receive a highly palatable food pellet. And so they'll spend some time taking the freely available chow and some time pressing the lever for the high effort reward. And basically, you can modulate their behavior with dopaminergic manipulations. So if you inhibit the dopamine system, they won't do as much high effort reward, they'll just take more chow. If you boost the dopamine system, they'll actually spend more time with the high effort choice. There's also a human correlate of this task, the human expenditure effort choice behavior. And you see the very same phenomenon. So if people are experiencing anhedonia or major depressive disorder, they will spend less, they'll spend more time with a low effort choice. If you boost the dopamine system, they'll also spend more time with high effort. So now you have an opportunity for translation and back translation of the rodents as well. And several other tests, which we also use. I won't go into all the details, but the splash test, light dark box, open field test, and force swing test as well. So with all of that, I'm not going to show this data today, but we did go and see and look at the effect of L-type calcium channels in some of these different behaviors, because again, these dopaminergic mechanisms are also important for behavioral responses to stress. And so when we took our animals through a chronic unpredictable stress paradigm, we saw changes in many of those behaviors, and we were able to reverse those with these L-type calcium channel inhibitors. So suggesting that these were not just decreasing relapse behavior, but also reversing some of the pro-depressive or anxiogenic things that were happening after chronic stress. But getting back to this whole concept of comorbidity, we also wanted to ask, well, what happens when animals are in cocaine abstinence? We know there's relapse behavior. Are there also changes in mood disorder-related behavior? And so basically, we took the animals through self-administration and then also through abstinence. And just showing you some of that data here, when we placed the animals in the elevated plus maze, both male and female rats, we saw a decrease in their open arm time. So again, this is suggestive of an anxiogenic response during cocaine abstinence. In the forced swim test, we saw an increase in immobility time, which people would associate with a pro-depressive-like phenotype. We also looked at social approach behavior. I'm showing you some of the behavior from females. You can see in saline abstinent animals, they do show nice pro-social behavior, spending more time with a stranger rat compared to the novel object. But during cocaine abstinence, that phenotype is completely lost. If we look at the splash test, basically in this test, the animals will be sprayed with 10% sucrose, and rodents will naturally start to groom after they've had that test. And we can measure how long does it take them to actually start grooming. So people use this as a very loose metric of self-care. So we see in the cocaine abstinent animals, there's actually a much longer delay before they start to groom compared to saline abstinent animals. And importantly, we also looked at locomotor activity in these animals. So all of these effects don't seem to be due to just general changes in locomotor activity that happen during cocaine abstinence. So giving us, again, a battery of assays that we can use to actually look at some of these behaviors. So what did we find when we actually thought about this whole L-type calcium channel regulation that I said was important for modulating relapse behavior, also important for modulating mood disorder-related behaviors after chronic stress? What happens after cocaine abstinence? One of the females, if we look at the open arm time, we again get that very robust decrease in open arm time during abstinence. If we give systemic administration of this low dose of the L-type calcium channel inhibitor, we can actually reverse that behavioral response. And we get a nice dose response curve with that as well. So suggesting, again, this is not only important just for the relapse component or response to stress, but other aspects as well. We did the same experiment in males, saw similar things, except we noticed there seemed to be a shift in the dose response curve. So with a low dose of isradipine, it didn't actually change the behavior. We had to go to a higher dose to see that same effect. So I mentioned this to say that it's very important that we actually continue to look at these sex differences, because if something like this actually translates to humans, it'd be very easy to make mistakes in terms of actually having clinical application by giving males and females the same dose of this L-type calcium channel. So again, I'm not going through all the aspects of the circuitry here, but we have done other experiments to look at some of these other aspects as well. And one of the things we're most excited about is the fact that we're now translating some of this work to clinical studies. So this has been actually a worldwide collaboration as we've worked with physician scientists in Austria, as we were working with the Laureate Brain Institute in Tulsa, Oklahoma, and using some of their research in that area, looking at longitudinal groups to actually start to think about these questions in those with comorbid substance use. It actually took us a while to think about how we wanted to design these experiments, because it would be much easier just to start in a population with substance use disorder or a population with mood disorders, but we really want to go after the relevant question as well. So this is a project that has been years in the making, but something that we're moving forward to in this year, and looking forward to moving things into the clinical population as well. The other piece that I'll leave you with is that we've also started to think about, as I mentioned before, some of these muscarinic receptor mechanisms as well, because the muscarinic receptors also regulate dopaminergic activity in the VTA2 pathway. And the M5 subtypes in particular have been shown to be localized primarily to dopamine cells, both cell bodies and terminals. So again, giving us some potential for therapeutic intervention. And one of the first questions we want to ask is whether these muscarinic receptor mechanisms also mediate some of these mood-related behaviors. So the first experiments we did, and Laura actually has been involved in some of this work that I'll talk about as well, but is to see what happens when we block muscarinic receptors in the VTA in the elevated plasmase in naive animals. Well, what we see is at highest dose, we can actually get, again, an anxiolytic response where they start to spend more time in the open arms when we give them this muscarinic receptor antagonist. Now, we also took some animals through this cocaine self-administration abstinence paradigm that I mentioned before, and we see very similar effects there as well. Again, the cocaine abstinent animals have a very sharp decrease in the open arm time, which we can reverse if we block these muscarinic receptors. One thing I hope you can appreciate from these slides is there's actually a shift in the dose response curve during cocaine abstinence, in that this lowest dose, 2.4 micrograms per side into the VTA, is able to reverse that behavior, whereas it had no effect in naive animals. So something, again, to be paying attention to and suggesting there's some type of cocaine-induced plasticity that's happening in the system. Albeit I should also mention that these animals at a 2.4 dose are no different from the saline animals, but it's still able to reverse that effect of cocaine. So the last kind of behavioral piece that I'll leave you with before I talk about some broader picture things is work that we've done, again, looking at this effort-related choice behavior and trying to understand whether these same mechanisms can also influence motivation. So in this instance, we again looked at the animal's behavior in terms of taking freely available chow or pressing a lever for the high effort reward. We set their lever pressing at baseline, and we found that if we enhance acetylcholine tone in the VTA, we actually get a pro-depressive-like phenotype. This is very similar to what people have seen in human literature for decades, going back to the 60s and 70s, where David Janowski and others actually saw that individuals who had been exposed to pesticides and organophosphates who had increased or boosted elevated acetylcholine tone actually had many pro-depressive symptoms. This actually led to the cholinergic hypothesis of depression. And we see very similar effects here in the animal models. So we increase acetylcholine in the VTA using the acetylcholinesterase inhibitor, and they decrease their high effort choice behavior. If we use a negative allosteric modulator of these M5 muscarinic receptors, by itself has no effect, but reverses the effect of the physostigmine so we can get back to baseline levels. Now, we also thought there's going to be a dopamine component that's likely contributing to this, again, because there's so much evidence about dopamine being involved in this process and because we're doing work in the VTA. Surprisingly, although I should say nothing should surprise us at this point, we saw no change in dopamine levels when we did these same manipulations in the animals that had undergone the same pharmacological treatment. Things were a little different when we looked at males, however. So we again do see a decrease in the high effort choice when we boost acetylcholine levels. No change with the negative allosteric modulator of M5, but now when we put those two together, we see a robust increase in their high effort choice behavior far above baseline. And we also see a dramatic increase in dopamine signaling in the accounts. Now, mechanistically, this doesn't actually fit to what we predicted. We would predict that if you have increased dopamine, you would have better high effort choice behavior, but we would not predict that blocking the M5 receptors on postsynaptic dopamine receptors would somehow boost dopamine signaling. So we still need to figure out what's contributing to this. But one of the pieces I think is really important here, again, is there are dramatic sex differences that are emerging between the male mice and the female mice. And so we need to pay attention to that and to also understand whether there are different mechanisms underlying the behaviors in the males and the females when it comes to these outcomes. So all that to say is we're continuing to do a lot of work in the system. We're actually now incorporating some aspects of vibrophotometry as well, where we can directly measure some of these acetylcholine changes, think about aspects of dopamine kinetics, and to continue to look at many of these different behaviors to really understand what's happening in the system. And hopefully, even with the muscarinic receptor manipulations, move those things forward into clinical trials at some point as well. So in the last part of the talk, I wanted to briefly highlight some of the other big picture things that I addressed in the title of the talk. Basically think about how do we actually take all this knowledge that we're developing in the lab and what do we do with that other than writing academic papers, which are good, which inform the field, which move things forward. But this image, which my daughter actually drew, was inspired by a conversation I was having with a hip hop artist. And from his perception, he said, it seems like all of you all in academics and academic medicine in particular, you have all this information and you've taken a bullhorn and you've pointed it at yourselves. And so you're just kind of talking to yourselves and nothing is coming out the other end. I took that analogy a little bit farther, and this is my perception of what I think people view us or our conversations like in the general public, that what comes out the other end is a bunch of nonsense that people can't understand. So I've tried to be very intentional over the years about trying to address this in different ways so that maybe we can reverse the bullhorn and actually turn it the other direction, and maybe we can say things that are actually cogent and helpful for those in the general public. And so there have been a few different ways that we've tried to do that. I mentioned to the students earlier, sometimes it's starting in small ways. So I think the very first time I talked about neuroscience in a public setting was in the 4th grade classroom when my kids were young, just talking about neuroscience at my kids' school. I also did things with kindergartners, doing the blind spot exercise. And so introducing these things really early on. You don't have to try and change the world all at once. You can do it one person, one individual at a time, and in really small ways. Another way, which I started early on, was with this STEM coffee hour. This was something that my wife actually started at a community library, bringing in different people at different times to talk about different aspects of science. So she asked me to do this. I really didn't have a choice. So basically I was voluntold. But wisely, I said yes. And she had me talk about the addictive brain. So this again was at a local library. It was a really powerful event because we were fortunate to also have some folks who were part of a recovery house who came to that talk. So they were able to talk about their recovery journey. And I went into this intentionally trying not to be there as a quote unquote expert. So I didn't want to be there just sharing didactic information, but really kind of create a space where we could have a dialogue. So a lot of those folks actually came in and shared about their own experiences with different treatment modalities, things that worked well, things that didn't work well, things that they thought the physicians may need to pay more attention to. The other thing is a lot of them came into that telling me things like, well, I don't see this as an illness because I did this to myself. Now, obviously at one level, that's true because you can't become addicted unless you use, but many of them also left that saying, okay, now I have a better understanding of how my long-term use is actually impacting my brain and how I can get to a new normal. So it's like they got more information, but then also had more empowerment in their journey moving forward. So that was a really, really powerful time. We also did an event at Yale in 2018 with that same hip hop artist, Lecrae. We brought him to campus. We partnered with the chaplain's office. We partnered with the vice president for student life. And we basically had a town hall conversation about topics at the intersection of mental health, faith, culture, and social justice. There were about 200 people in the room, but we created it in such a way that he could actually see all the people that he was speaking with. And it really was a dialogue back and forth, people who were sharing vulnerable aspects of their own journeys, even as Lecrae shared aspects of his journey. And there were students that came to that who said that was the most powerful thing they had experienced since they'd been on campus. So really having these conversations that cut across different disciplines and life experiences. We were also able to follow up with that with an event in New York City in 2019. Lecrae joined us for that. Vicky Sigworth, the National Alliance on Mental Illness in New Haven, also joined for that. Doug Middleton, an NFL player who had started a mental health advocacy and nonprofit after losing his best friend to death by suicide. They were all really vulnerable with their stories, sharing with the general public as well. We also had a second panel there where we brought in individuals from the community. Warren Kinghorn, who I see in the back, was gracious to actually join us for that and speak as a psychiatrist, but also as a theologian. We had other psychiatrists and psychologists who were there. We had community activists who were there. And there were about 800 people in the room, again, who joined and participated in that event. And then we heard people saying things like, I've been waiting for this conversation for 20 years. Obviously, we had to get a little bit creative in 2020. We couldn't have these 800-person gatherings anymore. And so we were able to continue some of these conversations online. We had a partnership between Duke and UNC, which I think was a miracle in and of itself. But we were able again to have that same conversation. Warren joined for that as well. And it was really nice to see the partnership there and how much people were really willing to live there, to walk into that conversation about anxiety in the midst of the pandemic. We also did some other events at other universities as well. Wesleyan University in Connecticut, having that same type of conversation. The one at Wesleyan was actually called a town hall conversation about race, faith, and mental health. So with all that, I also started to think about, well, how can we actually expand this conversation? People are saying they're getting a lot out of this once they're in the room. How can we make that broader and bring more people in the room? And so I shared with the students earlier, I did start this podcast in 2021. It took me some time to come to terms with it because I wanted to be behind the scenes and have other people run it. I basically realized if I didn't run it, it wasn't going to happen. So I eventually did lean into it and start this podcast where we've talked about a lot of different topics. Some of the topics that people say you're not supposed to talk around at the dinner table. The first one we did was called Wellness, Pandemics, Race, and Politics. So all the things you're not supposed to talk about, but we had a clinical psychologist and a political scientist talking about that together. And lots of different episodes about culture, about mental health in the military, faith and spirituality, being voiced for the marginalized. And these have been really transformative conversations that I've learned from some of these individuals as well, and that they've shared their stories and their experience. And so what I want to do in the last 2 minutes before opening up to Q&A is to show you 2 quick videos just to give you a sense of how some of these conversations have evolved and the types of things that people have been willing to share in those conversations. So the first one is from Dr. Alfie Breland-Noble, who's a clinical psychologist, and she was joined by Doug Middleton, who's an NFL player that I mentioned to you earlier. And let's see if the tech will cooperate. Hopefully folks on Zoom will still be able to see this. Let's see if I can shift things here. All right, so far so good. Is anyone on Zoom at the moment in the room? Okay, I mean, I hope the Zoom folks can still hear, but I'll check the chat in case they can. Go ahead and start this.
What I'd like about what Doug is sharing is definitely what shows up in the research literature, and that is normalizing it by showing people, reflecting people, giving people mirrors. So when they bring in multiple counselors or they bring in multiple people who are of color, right? What you're doing is normalizing for these young people. There are people out there who look just like you, right? Or look like your cousin or your people in them, right? Who do this work. And so those people, it's not always a perfect match, but they may be more likely to understand what your individual personalized experiences are, because there's universality to this issue, with this idea of, let's just keep it real, being Black at a predominantly white institution. I went to 3, right? My undergrad is from Howard, so that was heaven for 4 years. And then I dove out there into some of these other spaces and it won't so heavenly, right? It's kind of the opposite. And so even little day-to-day stuff, like we talk about microaggressions, you know, if you're an athlete and everybody knows who you are, that compounds it, right? Because everybody knows who the athletes are, especially if you go to a PWI, like one of my degrees is from Wisconsin. it was like 12 Black people there. And of them 12, 10 of them was athletes. And so, and if you weren't an athlete, people assumed you were an athlete because they had no conceptualization that Black people can go to college doing something other than, there's nothing wrong with sports. Like I said, my brother played college ball, but that's not the only way that Black people go to college. So my spouse, whom I met at Wisconsin, interestingly enough, was there getting a PhD in math. And he's kind of a heavy dude. And the people were like, yeah, you play for the football team? He was like, no, I'm here getting a PhD in math. And they would look at him. They were like, right, they thought they were getting pranked or something. And so that kind of stuff, you can only have that conversation with somebody who understands what that feels like. So no, I may not, as a mental health professional, understand what it feels like to be an athlete in that space, right? But I do understand what it feels like to be the only Black girl in the class. Right. And anytime a conversation comes up around anything related to race, everybody like this. Exactly. Right. Like, I don't have all the answers for everybody. Right. That's not what I'm here for. And so, what he speaks to is this idea of how do you normalize this concept of talking to people about what's going on with you. Stop internalizing it and holding on to it and thinking that you have to fix it all by yourself. So and that's in the literature. That's what we have to do with our young people of color. Other things.
All right, so a lot that you shared there, a lot of great information. Let me show you all one more video and then we'll shift to the Q&A. Hopefully I can make this work again. And this video is from, Ed actually alluded to this, from a Whole Man Project video series that I did that came out this past September. Again, integrating now aspects of mental health, psychology, neuroscience, and faith. So speaking primarily from a Christian perspective, but I think with principles that are applicable across the board. And just to give you a sense of how I try and tie all those different things together. Just make sure there are no messages that folks couldn't hear online. All right, I think we're good. Was the volume okay for folks in the back? Could you still hear the video? Okay.
Important. See, I don't see these as separate. Yeah. If anything, when I read the Word of God, I can see the ways that God has already given us tools in Scripture that impacts our health, our mental wellness, our psychology, as you were talking to. So even those things, we do have some autonomy to, as the Scripture encourages us to think about these things, but then also to know, as we know from psychological practice, like that starts a pattern. We can build those, quote unquote, build those muscles. Yeah. And that also impacts our brain. So the fact that we take the time to do what Scripture is guiding us to do, to think about these things, can actually impact how we think about those situations in different situations, different circumstances. And there's so many other examples of that. One that I often like to highlight is the passage in Hebrews that encourages us to continue to be together in community, so much so as we see the day approaching. I mean, you can easily, as a neuroscientist, see how God is calling us to community. And there's so much evidence from the research about the importance of being in community. I mean, just from the psychological lens, that's the first important piece. If someone is going through something, as you know, just to have someone to be able to talk to is the first step. It doesn't even always have to be someone who's trained in, being a counselor or a psychotherapist, but someone who just knows how to listen and to actually listen without speaking. So I think so many of us think we're listening, but we hear one thing and then we go on this long, different avenues about all the ways we're trying to provide help, just to sit back and to listen. And then just being in community, there's so much evidence that shows what that actually does to our brain. Just being in community with other people, as we are right now, actually impacts our brains. There's a molecule called oxytocin that's released in the brain when people are in authentic community together, when people feel like they're on the same team. It was actually discovered because it's a molecule that's important for contraction when a mother goes into labor. Wow. It's also a molecule that's important for connecting and creating that bond between mother and child. But it's not just mother and child, it's also the family relationship. The interesting thing, you actually see this amongst sports teams as well. So if you have a team that's going out against a common opponent, you see that level of oxytocin rise. It's also important in terms of how teams actually can create that bond amongst themselves, but actually how we all navigate in community. If we're going through something challenging, we actually see a rise in oxytocin that helps us be empathetic and supportive to one another. So I take that, say, look what God has shown us in Scripture about the importance of being in community, and look how the science actually backs that up and reminds us about how God has actually equipped us as human beings to gain from the ways we can be in community together as well.
That is so good, man. I was lost and confused, leading a council of fools. On my way to wholeness, I met some wonderful tools. Had a checkup with a doctor who's enthused to commune. Virtues all in tune, sitting in the waiting room. Love was talking to anger, justice talking to grace. It felt kind of fake, they all had the same face. Patients saw me in a rush, put me on the wait list, walked me to my seat and sat me next to greatness.
All right, so a little integration of neuroscience and music and hip hop there as well. And I don't know if any of you caught it, but that was alluding to this concept from Japan of kasugi, so something that's broken. It's actually filled with gold afterwards. It's more valuable than the original. So with that, I'm going to wrap up, get to the most important slide. It'll let me, maybe not. All right. And that's, of course, thanking all the people who have been involved in this work over the years. And this is just one small group of the, well, not small, but one team that was involved with lots of folks. As you can see on the other side, have gone through a lot over the years, but definitely want to highlight a few there. Eric Nunez, Dennis Baghdad, also Shannon Hughley, who was involved, Laura, who contributed some of this work as well. My L-type calcium channel collaborator, Anjali Rajadaksha, and my muscarinic receptor collaborators, Jeff Kahn and Dan Foster. More to come. As you can see, there's a lot of things that we're doing, but again, a lot of it has been informed by what we've been doing in community and what people are actually sharing with us as well. So hopefully I've got a little bit of time for questions, not much, but hopefully we can extend a little bit if you all have more questions.
We do have a couple of questions. Anyone, any of the students, trainees, any questions? Anyone? Is there anything online? So I had a question in terms of, oh, I'm sorry. Yeah, go ahead.
I was going to add one few hours or something to save it, but we're the all-time health experiments with the supplements region. And I have two questions.
Yeah.
One, have you looked at repeated exposure of this drug in next years? And the second is if these dopaminergic systems are convergent mechanisms of protein from plastics, have you tested whether or not this works with other drugs?
Yeah, so two great questions. We have plans to do repeat administration. We haven't done that yet. We've also been thinking about polysubstance use or other substances for years, and we just haven't gotten around to it in terms of bandwidth. So, and I'm trying not to make the lab bigger than it is, but it's always been that. It's a nagging question, so it's a great one. Hopefully at some point we will jump into that and actually address that. I do suspect it's going to be across the board, not just for cocaine, for other stimulants and other substances as well.
I had a question about, you have a question?
Oh, Oscar's first.
Okay, about the dark side, and things like stress-induced relapse. So things that might mimic the withdrawal from a drug might be kind of conditioned of like, oh, I'm going through this ill feeling. There, I know a drug would help. And so have you looked at that as well?
We haven't, and similar to the other one, just so many questions that we could address. So I mean, I've talked about that in some of the public settings, asked those questions, but we haven't addressed it from the outside. I do think there's likely both an L-type and muscarinic story there as well.
Sort of a similar question. When you mentioned that the L-type calcium channel inhibitors are already approved for hypertension, and showed really cool data, it made me wonder if there's a connection between the dopamine system itself, and I was doing a little Googling. I'm sure you know substance abuse and mood disorders are often precede diagnosis with hypertension. So just wondering if your lab is curious about that, like maybe dopamine to baroreceptors or
Yeah, that's a great question. Yeah, we haven't looked into that yet in terms of the dopamine and hypertension interactions. There's a lot to do there. One thing we try to do, which is different than your question, is actually look at medical records and see whether those with hypertension who were on different antihypertensives, whether there may have been different modulation of those mood outcomes. That project did not go anywhere because it was very hard. One, the physicians weren't necessarily recording that. First, we had to get access to records, and so the power just wasn't sufficient. So I know it's different than your question, but we have started to think about ways to do that. So we'll probably have to do it like you're mentioning going forward rather than retrospectively, which at the same time, but yeah, very good question. One in the chat?
Yeah.
So I can read out loud. Question about cues. If we see rapid firing of dopamine cells during cue paired with cocaine exposure, have you all done studies where you change the environmental cue, i.e. home cage versus novel environment? We haven't done that yet. Another good question. So all of you maybe should join the team. And I'm just kidding. But yes, definitely many questions that we could address in the future. And that's a very relevant and important one as well. I do think it would play a role there.
Good. Well, I want to thank Nii, Dr. Addy.
It's fine.
He wasn't a doctor yet when he was in the, he's a good time. Thanks for your attention and see you next week.